Our next presenter here is Dr. Davin Henderson. And Dr. Henderson received his PhD from the University of Minnesota, the other U of M if you're coming from the West, in biochemistry, molecular biology, and biophysics. He's a research scientist at the Prion Research Center at Colorado State University, and he works with Edward Hoover to understand the pathogenesis and replication potential of CWD in deer and elk. He has 5 years of experience in working with RT-QuIC, which stands for real-time quaking induced conversion assays, and 15 years of experience in protein biochemistry. He's authored over 10 manuscripts utilizing RT-QuIC and has pioneered the detection of CWD prions in excreta. So this has been brought up in several of the questions and interest in the audience already, or from Dr. Wood's talk. So if you would come up, Dr. Henderson.
Hi, everybody. Thank you very much for giving me the opportunity to speak with you today. I'm really excited to attend this forum because of the wide range of topics presented here. Usually when I'm going to a conference, it's mostly stuff like I'm going to tell you about, and I don't get the wildlife perspective, which I'm interested in. So I'm going to tell you today about primarily about the RT-QuIC assay and how we're using that assay to look at the early pathogenesis of CWD, as well as, is this advancing? As well as looking at the early pathogenesis of CWD and some methods we're using for antemortem detection, and then also a little bit about excreta at the end. So we know that Mary gave a really good introduction to the spread of chronic wasting disease around Wyoming. It's also expanding in North America. It's been recently discovered in Norway in free-ranging moose and caribou, so that's really concerning, and that those populations are fairly large, and then it's a new area for CWD. And the way we think this is happening is that there's this cycle of CWD where there's an event where either animals are contacted with the disease from individual to individual, or through contact with the environment. And through the disease course, this disease replicates, traffics certain areas, and then replicates. And then dissemination occurs through shedding of CWD prions in feces, saliva, and urine, and potentially through the spread of carcasses. So we're going to initially focus on, there's my topic slide, sorry. This is what I'm going to tell you about today. I think that was out of order. So early pathogenesis is the first thing I'm going to talk about. And but first of all, I want to give you a primer on what prion diseases are in the biochemistry, because the biochemistry of prion diseases is the basis of the technique I'm going to tell you about today to detect them. So prion diseases are found in lots of mammalian species, including humans, cows, sheep, and deer and elk, obviously. And the Crutchfield-Jakob disease is the human disease. Bovine spongiform encephalopathy, which was just a question, and that's the BSE or mad cow disease, which affected the individuals in the UK. Scrapie sheep is the longest known prion disease. It was the first one discovered, and chronic wasting disease is the one we're primarily concerned about in Colorado and Wyoming. And so what prions essentially are is they're a misfolded conformer of a normal protein in an animal. So this protein is in a lot of tissues in our body and highly expressed in the brain, and it has a normal function in maintaining potentially the homeostasis of cells. The full function is not really known, but this protein, when contacted by a misfolded version of that, can then be converted to that pathogenic conformation. So the cycle kind of looks like this. There's this sensitive protein that has a normal function in the cell. When an infection occurs, this infectious version has the ability to convert this to that infectious form, and then this elongates and fragments, and then this process kind of cycles like this. And it happens with in the cellular level, and then these fragments can break off and then be transported or spread throughout the central nervous system. In chronic wasting disease, there's a very strong lymphotrophic and early phase of the disease that's outside of the central nervous system. And I'm going to show you some new data from our lab on that next. But the cycle can be, I'm sorry. So the way these diseases are detected has been a really difficult problem for the prion field. So microbes like bacteria and viruses have DNA, which can be used with assays to detect the presence of those organisms. But prions are just proteins. So how can you detect the difference between a normal protein conformation and a misfolded conformation? So the detection assays have been based on the relative stability of this protein. So the prion protein is resistant to degradation. So in order to detect the prion diseases, there are a number of assays like ELISA and Western blot and immunohistochemistry which rely on that relative stability of the prion protein. So we can add an agent which degrades the host cell version. So if there's no prion disease present, you'll see no signal. But if there's prion disease present, then those resistant conformers will still be present, and you can detect the actual pathogenic conformation that's in that organism, in those tissues. And there are a host of new assays which rely not on the resistance of those proteins to degradation, but on the ability for those conformations to seed the new conformation. So it's a conformational-based detection. And these are RT-quick and serial protein misfolding amplification. And these are significantly more sensitive than the direct assays due to the fact that the amplification assays can detect the presence of these conformers by amplifying them to detectable levels. So extremely low levels of the pathogenic conformation could be amplified by adding a sensitive substrate to that sample. So if there's a prion present, it will convert that substrate to more prion-like protein, and then we could detect that converted product. So it's sort of like protein PCR, if you're familiar with PCR. It's a way to amplify the signal from undetectable to a detectable level. And that's what I'm going to tell you about today. And RT-Quick has been shown to be about 10,000-fold more sensitive than ELISA or Western blotting. And the basic mechanism is sort of like what I said. We create a PRP monomer in bacteria, which is a sensitive protein for conversion, and we add a sample to that substrate. And if that substrate has that prion amyloid conformation, then that prion amyloid will seed conversion of that substrate, and we'll get, through multiple rounds, we'll get an amplification of that product to a detectable level. So, and this is also a fluorescence-based assay. So there's a dye that specifically binds to this amyloid conformation, and as that amyloid conformation increases, you see an increase in fluorescent signal. So that was a lot of biochemistry to start off the talk. I realized we just went from the big 40,000-foot wildlife perspective down to the very smallest level of prion disease. But that's kind of what I needed to get out in order for you guys to understand the rest of the talk and understand this new version of detection versus these direct detection assays and how these can potentially benefit the chronic wasting disease research. So the other good things about this assay is it's between 24-hour and 60-hour reaction time. Some of the other amplifying assays, like PMCA, can take multiple days or up to 7 days. It's sensitive and extremely specific, greater than 95% specific in any really well done studies to date. The data are in real time. This is what they look like. So we add the samples at the beginning of this assay. It goes into this 96-well plate, into this fancy shaker, and through time, we can see an increase in this fluorescent signal, and this is a CWD positive sample. It's also really easy to optimize because of the 96-well plate format, and we've developed a number of extraction methods to take to extract or concentrate CWD prions in certain sources like saliva and urine, where they're at very low levels, and then to allow for detection in those. So those samples are even difficult to detect with this assay. That's extremely sensitive. So we use this assay in a study. Claire Hoover, not related to Ed Hoover, but she's a DVM PhD student in Ed's lab, and she's been looking at this early step between entry and trafficking and the early sites of replication. So this is unpublished data, and this manuscript is, I think, submitted, if not on its way. So this is the study design that she had. So early infection from the point of inoculation, she followed deer at 1, 2, 3, and 4 months post-inoculation, and then looked at a number of lymphatic tissues, so lymph nodes like the ones in your neck, for the presence of CWD by 2 assays. The RT-Quick assay, so one of the most sensitive versions we've been able to develop, and then also IHC. And she looked at multiple genotypes at these stages as well. So 3 GG deer here, GG, and then a handful of GS deer. Those are rare in the animals that we get, and the GS deer are equivalent to the ML elk. So the S makes them, the GSs are more resistant to disease than the GG. So, and I'll show you some data on that. So at 1 month post-inoculation, she was able to see the beginning of a signal here. And so these are the data I'm going to be showing you today. This is a RT-Quick reaction rate, which is a little bit different than showing you a bunch of those lines. The lines get pretty confusing. You get too many on one graph. So this, the higher the symbols are on this, it means the more CWD prions there are in the sample. And down below, there'll be a number of tissues or different samples, depending on if it's excreta. And the different colors here are different animals. So you can read into these data as much as you want and scrutinize them, or you can just kind of get the general trend by watching the symbols go up as disease progresses. So in 1 month, there's a small amount of signal starting, but not particularly well advanced. But at 2 months, I'm sorry, so these are statistically different than the negative control animals. And at 2 months of age, you can see that there's a significantly increased level of, should have the bars, here we go, a significantly increase in signal here, and specifically in the tonsil and in the retropharyngeal lymph node here. So just after 2 months of infection, we can start to see CWD in these lymphatic tissues. At 3 months of age, you can see that now throughout the draining lymph nodes of the head and neck, there's a significant accumulation of CWD prions in these samples, and all of these boxed are different than control. So just after 3 months, there's a fairly established replicating population of CWD prions in the lymph nodes of the deer, and these are whitetail deer. So at 4 months of age, there are a similar trend. So if you kind of get the difference between 3 months to 4 months, you can see that the numbers are increased. So that's good evidence that not only is our assay detecting the increase in prions as disease course goes on, but that these numbers are actually following a disease course and not some erroneous type of from our assay. All right, so I showed you the 1 month data from the GG deer here. And this is the summary data, so this is where the numbers kind of increase in a very nice trend. So there's very little infection at 1 month, and they get bigger at 2 months, and they get bigger at 3 months, and they get bigger at 4 months. So the only difference between these dots and the last ones is these are the average data for all 3 deer in the group put together. So it's kind of a nice summary to see the trend of infection increase and then become more widespread. And some of these levels at 4 months in these lymph nodes are equivalent to the terminal levels in animals that have died from CWD. So at 4 months of age, the accumulation is fairly substantial in those lymph nodes. And to look at the IHC to mirror the RT-Quick data, the same samples that were RT-Quick positive at 3 months were also positive in the tonsil and the retropharyngeal lymph node by IHC, and then the tonsil and retro by 4 months. You can see an accumulation there in the increase in the staining. So the red is the CWD prions, and this is what I was talking about, a direct detection assay. So a protease has been added to this to remove the sensitive protein, and the only red staining left is the CWD prion present. And the important thing about this, all of these deer tested, is they were completely negative in the obex, or the brainstem of the animal. So this infection is prior to neuroinvasion of any kind. So after oral infection, the lymph nodes of these animals are extremely positive prior to neuroinvasion. So we think that might have something to do with the shedding of CWD prions earlier in disease. The tonsils are positive very early, so we think that might contribute to the shedding in saliva. All right, so to summarize these data by IHC, if the RT-Quick results were a little bit more involved, the IHC results are summarized nicely in a table here, where you can see that at 1 month there were very few lymph node samples positive. The tonsil is positive very early. And then as infection increases, there's more and more IHC positive tissues, which correlate very nicely with the RT-QuIC results. All right, so if we take a look at these animals, the GG animals compared to the GS animals, we see a slightly different picture. So this is the 1 month for the GG, where we're starting to see a little bit of seeding activity. And in the 1 month GS, there's almost nothing. So these look nearly identical to the negative control animals. Now looking at the 4 month GS deer compared to the 4 month GG deer, we can see that we're just now starting to see positivity in the tonsil. And importantly, the tonsil was also the first tissue that was positive in the other animals. So the disease is not progressing differently in these animals. It's just progressing slower, we think. So we haven't had many SS animals, which would be the most resistant version of the genotype. And we're interested in trying to look at some of those. We have one that I'll tell you a little bit about. Oops. Right, and switching. So this is an SS, the one SS animal that we had at 16 months post-inoculation, and it was positively inoculated with an experimental dose of CWD, which is much larger than an animal would get in nature. So the likelihood of it being infected was fairly high. But even with that, the disease progression was significantly slower than the other GG or GS deer. So these are a little bit smaller, the 3 month and 4 month GG deer compared to the 16 month GS deer. And these are all on the same scale. So you can see that this SS animal is kind of equivalent to maybe between a 3 month and a 4 month GG animal. So at 16 months of age. So significant slowdown in disease progression in those SS deer. And to summarize those here, this is the 16 month IHC data. And compared to 2, 3, and 4 month of the GG data. So pretty much equivalent to the 3 to 4 month range, which is kind of amazing that it's delayed, fourfold almost compared to the GG deer. Oops, I took out the summary slide. So in conclusion for the early infection, we see that the lymph nodes of the head and neck are affected early in disease, and those are very positive, almost to terminal levels prior to neuroinvasion. So now I'm going to tell you a little bit about our efforts to use RT-QuIC to determine the antemortem CWD status of deer. So these are a couple of papers that were recently published using the RT-QuIC assay, and currently real-time RT-QuIC is being used to diagnose sporadic Creutzfeldt-Jakob's disease in humans. So there's a lab in Cleveland that's using this assay currently in the clinic for doctors to send CSF samples to determine the status of humans with this disease. So it's been validated by, I think there were 4 different centers in a manuscript to validate the technique with the same samples, and they all got the same answers. So it's a very reproducible assay in multiple labs' hands, and with those data, they ended up making it a human-based test. So I think we could use it to determine the antemortem status of deer and elk using the rectoanal mucosa-associated lymphatic tissue, or RMALT. It's basically a rectal biopsy, and it's fairly well tolerated in animals, and it's easier to take than a tonsil biopsy. However, I just showed you that the tonsils are positive significantly earlier than the RMALT. So the ability to determine if an animal is CWD negative with RMALT is slightly reduced compared to tonsil, but it's still a good indicator and for a high throughput screening of lots of animals, for example, in a captive facility, it may be easier to use RMALT samples. So what the sample looks like, I know you're all really curious about that, is right around the interface between the inside and the outside, there is this ring of mucosa, and this sample is a lymphatic tissue similar to the lymphatics found in other parts of your body. And this can be taken out and snipped off, and a sample be tested by RT-QuIC or even also IHC, the gold standard assay for prion detection. So this has been used in sheep for quite a long time to detect the presence of scrapie, and it's been used quite a bit by Nick Haley in a couple different studies, as well as a project that I'm involved with Nick on, working out here in Colorado on an elk ranch that has a captive herd of about 450 animals. So Nick had 2 manuscripts where he looked at the remalt samples from both elk and deer, and he found that there was about a 96 or 95% correlation between the RT-QuIC assay results and the IHC assay results. And in the current study we have for elk at this ranch, there were 34 total IHC positive elk and a total of 62 positive by RT-QuIC. So the assay is likely more sensitive than IHC in detecting animals that could potentially progress to IHC positivity. And this version of RT-QuIC that we did here is significantly less sensitive than the one I just told you about. So this is a simple 24-hour protocol with just a sample dilution. So this has some area for improvement. I think we could do a more sensitive technique and find even more positives in this herd. And one thing we're interested in with this herd specifically is understanding the difference between an RT-QuIC positive animal and an IHC positive animal. So we know that IHC positive animals progress to terminal disease. So do RT-QuIC positive animals also progress to terminal disease? Or is there some way that they can clear the disease if they become positive? No one's ever shown that you can get rid of chronic wasting disease, and we don't know that either. So, but if we can detect these really low levels in the animal, do they stay low and never go up? Is that possible in the animal, or can they actually clear the infection? We don't really know. I don't have a lot of high hopes for that, but it's kind of something we've been hoping for in this herd. So we're going to test those 450 samples again with a more sensitive technique. The same story in deer. We saw a total of 128 positive by IHC and a total of 195 by RT-QuIC. So again, there's an increase in specificity. Increase in sensitivity for the RT-QuIC assay over IHC, and this assay, the specificity is also excellent. In these two elk studies, there were no not in this elk study because we don't know the negative animals, but the human studies, it was 100% specific in the two human studies. In a number of CWD studies that we've done, it's been 100% specific, so never detecting a false positive when statistics were performed on those data sets. So the way we kind of see this working is primarily helping out captive herd populations and managing CWD epidemics in a closed system where they can look at the CWD prevalence and maybe target certain animals for removal to keep the prevalence down compared to the outside area. Obviously, this would be in an endemic area. If it was in a non-endemic area and they found CWD in a captive facility, there might be a different management strategy put into place. But what we'll see is that there are a certain number of IHC positive animals which could either be culled or followed closely for clinical symptoms. And then there'll be another series of RT-QuIC positive animals in blue here. And then those animals may progress to IHC positivity eventually. So as a manager, you can decide, do you want to try and get rid of CWD in total, potentially, or just get rid of the animals that are likely to develop disease in a shorter period of time? So we're trying to give people options besides complete depopulation, as well as give a more robust picture of what the CWD status in the herd or group of animals might be. So, like I said, remalt is a good sample for detecting CWD. It's not the best. So these are data from the spleen of the animals I showed you before, and the spleen is very positive. Early infection from up to about 100% in early infection, and the remalt is not quite as positive early in disease, but it still is detectable. And at terminal disease, they're identical. The CWD is detectable by both IHC and RT-QuIC at terminal disease. All right, so I gave you a little bit of introduction about how CWD is trafficking early in replication and some ways that we can look at antemortem detection of CWD. Now I'd like to talk to you about how the prions are coming out of these animals and potentially contaminating the environment in a new series of tests we're pretty excited about in being able to assay the environment for CWD contamination. So we had some previous work a few years ago using the RT-QuIC assay to detect the presence of prions in saliva and urine of white-tailed deer. And these are captive white-tailed deer that are experimentally inoculated with CWD. So the dose is fairly high. The levels of contamination or presence of CWD prions in a more natural infection is something we're still working on. So the doses we give establish infection quickly, and the reason we do that is because it's very expensive to keep deer around for a long period of time. So doing a low dose, longer term study is extremely expensive. But those are probably the studies we need to be doing to really get a good picture of the real potential for CWD shedding over the lifetime of an infection, because it's a longer time period, and potentially there may be we have some very preliminary data that suggests there may be some more shedding in animals with a lower inoculation dose, but we're still parsing that out. But this is summary data from I think 6 different deer, and you can see that throughout the disease course, the detection is not particularly high, so about half of the samples at any given time could be positive. If these were individual animals, the pattern would look a little bit different. So what we actually did see is that we'd get detection at 3 months, and then not detection at 9 months, and then detection at 12 months, and then maybe detection not until 21 months. So the pattern is up and down for shedding with these animals, and we're not sure why that is, if it was a failure on our part to actually detect the prion, or if it was the fact that the deer was actually just shedding less prion at that point. We don't know the answer to that. We have a graduate student, Kristen Davenport, who's doing a modeling study. We've tested all the saliva samples from all 13 years of deer we've had, with about 12 or 13 deer in each group, and now she's trying to model our ability to detect CWD prions in that population of saliva and determine what the problem is. So are we missing samples, or is it actually intermittent shedding? So hopefully the model can answer that question. I'm not a modeler, so it's a little bit above my head. But in urine, we see a little bit, kind of the same story. We don't have as many urine samples as we do saliva samples. The male deer are a little bit easier to sample for urine than the female deer. Catheterization can be tricky. I've never done it, so I've collected the feces and the male urine, because the way you do that is you tape a bag to them and they wake up, and you follow them around with the bag on until they go. And I was the guy who followed them around looking, and you have to keep them from taking the bag off. So it's quite a rodeo for a little while there. But you get great male urine samples, hundreds of mils, which is great. So that's why these data are not quite as fine as the saliva data. But we do see shedding in urine as well. But what I'd like to point out is on here, these are our estimate at the levels of CWD prions in these samples, which we think is fairly representative, but we haven't been able to confirm these with bioassay yet. But we kind of see a trend where later in disease we see higher levels, so not until you get more terminal in disease, and also the saliva seems to be significantly higher than the urine in most cases. So. And this is, I actually took that picture. So the thing we're most excited about recently is we've been able to detect CWD prions in the feces of both deer and elk. So the method to do this it took a few years in development. The feces has the potential to not only inhibit our reaction, but also cause spontaneous conversion of the reaction. So it was kind of the worst of both worlds. Decreased the reaction temperature, which decreased the false positive rate, and then we also used an extraction technique which kind of got rid of some of the inhibitors in that in that deer or elk feces, in order to kind of clean it up, because it is a little bit dirtier than saliva and urine. In our captive animals, we saw a prevalence of approximately 50% at 12 months after infection. So feces doesn't appear to be an early marker of disease. It's more of a late marker of disease. But in any case, the level of detection at later in disease is approaching 80%. So we feel like if there was a deer shedding CWD prions in feces that was near clinical disease, we would probably be able to pick that up most of the time. So that has the advantage in that feces you can actually find in the environment, versus urine and saliva are not exactly things you can go out and get. But feces is fairly easy to find. I found it right here at Pingree Park. I took that picture right before a talk, and we're working on determining the level of degradation of the signal in aged feces versus fresh. But in a large herd, like the one we just saw out front, I'm sure there are fresh samples that are easily accessible to go out and test. which has a couple of advantages. It's very easy. There's no need to go out and target a specific animal or knock them down or use a dart or but you could just kind of get a picture of what might be happening in a herd or in a landscape without having to do a lot of animal wrangling. Which is good for me because I'm a biochemist and animal wrangling is not my forte, but picking up feces would be fine. So to look at these data a little bit finer level, so these graphs are similar to what I showed you before, where the higher the numbers, the more CWD prions are in the sample. And this is a time course of infection from our experimental animals where they're sampled at 3, 9, 12, and various months post-infection. And the three different colors are different temperatures. So with decreasing temperature, we saw a decrease in the false positive rate, but these are the positive animals, and you can see that in each temperature, we don't really see a decrease in the positive samples. So the negative samples seem to go away when temperature decreased, but the positive samples remain. So we feel these were bona fide CWD seeding activity versus this activator that we found in these feces samples. And this demonstrates nicely the difference that different animals could have. These animals all received the same dose of CWD, but this animal at the top, at 9 months post-infection, developed a fairly substantial shedding of CWD prions in its feces, where this one didn't really see anything until 23 months post-inoculation. Why that is, I don't know. I guess these are also not congenic animals, which means they're not identically related to one another like we have in other laboratory animals. So there's various genetic factors which could potentially affect this. And these are all GG animals, so it's not the resistant genotype, which sheds later. And 816 here is more equivalent to 813. We're trying to test more animals in our repertoire that we've saved feces over 13 years. We have a whole freezer full of it. So we're on our way to testing some of those samples to see if we see more patterns. We've also done this in the same elk samples that I was telling you about in Colorado. The elk ranch, when we collected the remote biopsy, one of the side effects of collecting a remote biopsy is removing feces. And I was there to catch the feces, and Nick was there to take the remote biopsy. So we had a team going on at one end of the animal where the other people were taking blood and saliva swabs and other things at the other end of the animal. And the ranch owners were doing their deworming and ear tagging, and it was quite a process. So these elk are run through a squeeze chute, which the squeeze chute operator is the single most talented person I think I've ever seen run a mechanical instrument. That guy could really manage elk going through a chute, and it was really fun to be involved with. But we did get feces samples from 24 of the RT-Quick positive animals. We didn't get feces from every single one of them. And these are the results from animals that were RT-Quick negative in the remote biopsy. We don't know if these animals are truly negative because they're on a ranch with CWD. But they were RT-Quick negative, so we used those as negative feces samples. And we ended up seeing quite a bit of positivity in these elk. And the important thing here is that these are preclinical elk. None of these elk were exhibiting overt signs of disease when they were going through the chute. Otherwise, they would have been tagged as so. And a number of them were IHC positive as well. So we saw about a 40% detection rate in IHC positive elk. So the elk that were IHC positive in their remote were also about 40% of those were positive in their feces. So again, not the best predictor of disease, but potentially a sample that we can use to look at environmental contamination. All right. So to conclude, I've told you about prions at the very beginning of disease and how we're using our RT-Quick assay to kind of push the limit on where prions are found first and at what levels. We've also shown you that the resistant genotypes have a delay, but they don't stop replication of the disease. And the SS animals still do get chronic wasting disease. And we're developing this RT-Quick assay potentially as a next generation assay as antemortem testing for CWD, and we're hoping to move forward with that, not only with this elk ranch, but with the Wisconsin DNR. We're potentially collaborating with them to get some feces samples from a very large study that they're starting in the near future. Feces and remote samples from that study. We're also hoping to use it to apply the RT-Quick technology to environmental samples to determine the presence of CWD prions in the environment, as well as potentially the level of CWD contamination in the environment. We're starting to test not only the feces, but scraping certain objects that we can find in a positive environment, like a salt lick or a fence post or something like that, and also soil samples. We're both experimentally and naturally looking at soil and its ability to harbor CWD prions using the RT-Quick assay. So I think that's all I have for you today. So Ed Hoover's my boss. He's been a fantastic mentor and a really, really good boss. He actually couldn't be here today. That's why I'm here. Joanne Tennant is my graduate student who did a number of the feces experiments, actually almost all the feces experiments. Kristen Davenport, I was talking to you about, did some of these, the modeling stuff that I was telling you about. Nate Denkers has been our deer wrangler in-house, doing all things related with the deer, and also developed the magnetic, or the iron oxide particle concentration method we use for the feces extraction. And Claire Hoover did all of those early pathogenesis studies. And Nick Haley is my close collaborator who we're working on with RT-Quick and CWD. So with that, I'll take any questions. Thank you.
Okay, well, that will be the 5 minutes for questions. So, Dr. Henderson, sure.
Right here. There's one close by.
Well, thanks for all that. Much of it was way above my head. So
I was trying to go both sides, but
looking at the RT-Quick methodology and in all the different samples that you use, I could see muscle tissue on the
right.
list. And I wonder if muscle tissue is detectable. And then looking at some of the control methods that we see for transportation of carcasses, is limiting transportation of carcasses, eliminating spinal column and brain matter, is that maybe the best strategy? And then the last part of the question, very long question, is knowing everything you know about CWD, would you consume meat from a CWD positive
right.
individual?
Okay, so do I have to repeat the question?
Yes, please.
Okay, so sorry, I didn't mean no offense about repeating. So are there CWD prions detectable in muscle, and would I eat that muscle? I think is the summary of the question. So we've done a lot of studies just recently looking at a real sensitive survey of all the tissues positive in a terminal animal, and various muscle tissues have been positive. Like we found a very positive heart tissue. I've tested other muscles that are not detectable, so it seems to be a little bit more iffy in muscle than other tissues. Lymphatic tissue, 100% in terminal animals, every single little bit, and you can't really take out those from the carcass. I mean, you can remove the spinal column, you can remove the head, but there's still lymph nodes spread all over the animal, which have potential to infect animals at a pretty high rate. In experimental studies. Would I eat CWD contaminated meat? So I work in a lab where I can't leave with the lab coat that I put on inside of it and have to be wearing gloves and goggles when I go into it to just touch the stuff that was touching the CWD. So it would be shocking to even think about consuming that anything in that lab. So in general, no, I wouldn't. And especially as a biochemist, we've used the human prion protein to convert CWD prions, and those two have no problems getting along. The human protein converts extremely rapidly in the presence of CWD prions. The CWD prions put into a humanized mouse, however, is very resistant, extremely resistant, more resistant than a lot of other transmissions to replication. So an infection does not get established in an animal, but biochemically there's no reason why it can't happen in our in vitro system. Oh, geez, sorry.
I think that work offers tremendous possibility for
Thank you.
understanding effects of CWD in populations by giving us an assay for the environment. But I caution whether or not we could ever use that assay, at least without some additional work, to determine whether animals, the infection status of an animal, simply because most of the dry matter in feces is exogenous in origin.
Right.
It's undigested plant residue and soil. And so we can only talk about that animal if we could somehow separate out the endogenous
Right.
portion for that assay. Are you working on that? Is
Right.
that a problem you're up against? And you know, that was a long question, so maybe you don't have to repeat it.
I think I can summarize. So, am I or are we concerned about pass-through of prions? So if an animal is eating in a region where there are prions in that environment and it passes through in the feces. So what we're doing to look at that is actually looking at the environment to see if we can detect CWD prions in those situations. Now, the environment is very large, and the presence of those prions they don't raise their hand when you walk by, so it's difficult to know where we need to sample to get those type of samples. But from my estimation, the consumption of a let's say there was urine or a saliva consumed, and that gets diluted through all the different stomachs, and then through, I think that the dilution factor and the dose that would be required to see the seeding activity that we're seeing in feces would be unattainable in excreta. So we can try to model that in the lab by putting that out and then diluting it to the point where we think that the insides would be equivalent to, and then testing that. So I think it is coming from through swallowing saliva and through a pass through in the digestive tract. So yeah, we can't rule that out. But the main thing we're really seeing that I think we could use that for is that it's not that is that animal sick, but is there CWD present? Period. So in an environment where there's not a lot of money for testing and they want to go out and sample it and potentially see if there's CWD there at all, and they find feces piles and they could pick 100 of them up and we could test them all. And if we find 4 or 5 that are really, really positive, the likelihood of there being CWD in that environment is also, I think, fairly high. And that might warrant better analysis of that specific area. So that's kind of how I see it being used, not as a specific animal marker. In the middle, I think you've had your hand up. Here.
Is there any data on how long shed prions stay active on the ground?
On the ground.
And then just following on, just out of curiosity, what the heck do those elk farms do when they find they've got infected elk?
Right.
They shed prions, and is there any chance those things could end up in the food chain being sold to
Right.
restaurants?
Right. So how stable are prions in the environment? Joel is going to cover that in the extent, at a much more extent than I could explain, but the answer is a long time. Prions have been shown to be active for decades in the environment. So take animals out of an area for years and years and years, put them back in, and they'll get sick from that same environment without any animals being on it. So the answer is a long time. I don't know how long. Maybe Joel's going to tell us. That'd be handy. What was the second question?
What do the elk farms
Oh, elk farms. So what do the elk farms do when they find out that they're positive animals? And the answer is it depends if it's a male or a female. They decided to call all the IHC positive animals that were female, and the bull elk they kept because it was a shoot farm, and those are extremely valuable animals to them. And we can't tell them what to do, especially I can't tell them what to do. I can just tell them the results of the tests. So those animals we determined would likely would take care of themselves, but it's at their risk that they leave them on the environment. And can they get into the food chain? I don't know what they do with those animals that are hunted on that ranch, whether or not they consume them or not. I don't know who does. I doubt the people coming in to shoot elk for $30,000 are taking it with, but I'm not sure. I could be wrong. Yep, over here.
So if I understood you correctly, for these resistant elk, with the most sensitive tests, it could take up to 16 months to detect a CWD positive animal. And if that's the case, does that mean that we have a number of infected individuals on the landscape while we're doing these surveillances across the state that aren't being detected that are purely positive?
Yes, I think so. The question is, with the resistant genotypes, is there the chance that you'd be missing positive animals due to the testing regime? So an IHC positive test or an RT-Quick positive test, I think tell you two different things. The RT-Quick, I think, gives a good indicator that animal will progress to an IHC positive state. And is that animal going to have CWD? We think so, and we're working on that now. But the length of time needed, I guess it's two different circumstances. In a wild animal case, I think there could be a significantly different prevalence. One study that we did using PMCA, actually, which is the other amyloid amplifying assay, on fetal tissue on an elk population, there were, I think, a prevalence of about 7 or 8%. I can't remember the exact level, but it wasn't above 15. And in the PMCA samples from those same elk, the prevalence was probably closer to 30 to 40% after all that analysis was done. So using the most sensitive tests, the prevalence is likely a lot higher. But are those animals, dangerous to consume, or, will they always develop disease in total? I don't know.
We have time for one more question. We have just 1 minute.
Right over here.
Yeah, with all the amount of CWD in Colorado, from your any so-called members of L, is there any cases in the medical field, and the hospitals, your doctors, your scientists, blah, 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 have any cases showing up with the hunters actually coming up with being sick by CWD?
Currently, there hasn't been a case linked to consumption of CWD positive meat that I'm aware of. We did get some funding recently to look at appendix samples from individuals in Colorado. So there was a study in the UK where they found that appendix samples from people who were exposed to the BSE, the mad cow epidemic, and tonsil samples from those people were positive at a rate of about 1 in 20,000. Those people don't have disease, but they are positive for mad cow disease. And I don't think they told them. Maybe they did. I don't know. But so we're doing a similar study in Colorado to see if there is a subclinical level of prion disease present, but we don't know the answers to those studies yet. Thank you.
We need to thank Dr. Henderson for coming to see us.
Thank you.